Okay, ma'am.
Can you see that recording option?
Yes, ma'am. Start it, ma'am.
Okay. Dear friends, today we are going to discuss more about nucleic acids again. What we have discussed in the previous revision session, again a few points we will be going through, and then we will enter into the next part, cell division. As we have earlier seen, the nucleic acids are made up of all nucleotides, nucleoside, and at last it is with nitrogenous bases and hydrogen bonds. It is all tied up together with a sugar phosphate backbone at the end. So all these things we have discussed in the previous session. And the very first step, how it is being formed, is formation of a nucleoside. Here I have marked one carbon in the ribose sugar. That is the first carbon. So the counting starts from the clockwise angle, 1, 2, 3, 4, and 5. The first carbon reacts with N of the nitrogenous base, and that forms the first reaction, nucleoside, and thereby one kind of setting started in this synthesis. The very next step is to form a nucleotide by bringing the phosphoric acid inside, and this moves one water molecule outside, and a nucleoside is getting converted into a nucleoside. This picture will show you exactly clear way of representing it. Like normally we write the sequences from the 5' end to the 3' end, where we do have this nitrogenous base at the one end, and that is connected to the first carbon, and this is the second, and this is the third carbon where the next nucleoside is going to get added. It can be a nucleotide, or after that it is directly a nucleotide also, means whatever it is, the incoming molecule is getting added to this one. And here it is, this part is connected with the 3' end of the previous molecule. So it runs from the 5' to 3'. This normally we write like this, 5' to 3' only we write. So you just remember these molecules, the first one, first carbon, and the second one is with the hydroxyl group, and the third one, here it is running to the next molecule, and this is the fourth one, whereas this is fifth one that is connected to the phosphoric acid, and at last this is getting connected to the previous nucleotide. So just remember this structure. Whatever question comes, you can easily write. You can easily answer this one. So we know that this is running in one direction. Like we know this. There is one direction, one kind of strand we have seen, and the opposite strand runs exactly anti-parallel. That means the 5' end of one strand is getting complementary paired with the 3' end of the other strand. So that means like two buses running in the opposite direction in the same road. It is like that. Okay. So next one is, we know the structure of DNA. We know the structure that is inside towards, means based on an angle, means a fixed axis. It is all stacked like stairs, helical structures, and at last there is a sugar phosphate molecule, and this forms major and major groups. All these things we have discussed in the previous session. So if you want to recollect, you can just go through that once. And actually, just because of the presence of van der Waals force and the covalent bonds, the inside of the molecule means van der Waals force acts inside the molecule, in between the stacks, and this makes more of hydrophobic in nature. So inside the DNA helix, it is hydrophobic. The outside is hydrophilic one. So outside sugar phosphate backbone is there, sugar is there, sugar is highly hydrophilic, means just you keep your sugar box open in the kitchen and the very next day you can see it is very sticky just because of it is absorbing water from the atmosphere. This you can keep in mind so that the outer part with the sugar phosphate backbone gives you the hydrophilic property of the GNA. So that is the shortcut to remember the thing. It is not exactly that much hydrophilic just like your sugar, but it is relatively hydrophilic when comparing the hydrophobic inner core of the DNA helix. Okay, so the anti-parallel nature of these two strands define how this molecule is going for replication process as well as the transcription process, because all these strands have different functions and different places it is acting differently. So we know that it is all composed of a nucleoside, and nucleoside is having a basic unit, a nitrogenous base. And how this nitrogenous base is synthesized, that will be a very sugary question. It is very sweet just because the purine synthesis starts from a sugar molecule. Okay, so you just remember like purine synthesis is so sweet, and that starts from a sugar molecule, ribose sugar. Okay, so that is the easy way to remember all these things. And just this picture gives you, means everything is in your book. We are just opening your book. Whatever slides I'm showing here is exactly from your textbook, what we recommended in the classes. So just go through your textbook and just open this page. You can see this, like what are the molecule, means each and every atom is coming from which molecule. So everything is very clear. Everything has been tracked, how these purines and pyrimidines are being synthesized in de novo, or whether it is being recycled, whatever it is. So normally, all these active synthesis in animal system takes place in liver, whereas in plants, the already matured or completely developed cell don't need to produce all these purines and pyrimidines by wasting a lot of energy in doing that. So what actually is happening is the actively dividing cells of meristematic regions, they do a lot of purine and pyrimidine biosynthesis. And this you should keep in mind, and it is all being regulated, means enzymatically as well as hormones also play some role. There are a lot of regulations that we will see in the next few slides. The very first step of purine synthesis is, I said like it is a sweet pathway. So just remember that it starts with ribose. So just like your sugar phosphate backbone, we say everywhere it is sugar, sugar, sugar. So just everything is starting with ribose again. The same sugar is here. Okay. So this ribose 5-phosphate again takes phosphate molecules from an ATP, and it takes 2 phosphate molecules, and then it leaves A and B. Okay. So now it becomes a pyrophosphate added to this end. That is the first carbon. Okay. So you just remember that a ribose phosphate with a pyrophosphate. Okay. So phospho, ribosyl, pyrophosphate. So this is the compound. It is so simple. This is phospho, ribosyl, pyrophosphate. So this is the actual first step, that is PRPP synthetase is the first enzyme actively involved in purine biosynthesis. But in many books, it is mentioned like the next one is the first step, means this molecule getting converted into an amino molecule. Okay, so that means here it takes glutamine in, and then an amino group is added here. So this is normally mentioned as first reaction in many of the books, but irrespective of whether it is mentioned as a first step or as a second step, The last one is the same, that is inosine monophosphate is the last molecule from this purine biosynthesis. So many books says like there are 11 enzymes and 11 steps involved in this pathway, and some says it is 12 and 12 in 12 enzymes. What we are seeing here is actually the interconnectedness of all these things. Okay, so the intake of urates or the purine alkalides play a role in this one. The levels of energy in a cell means if the cell is having a lot of energy to produce new cells, then automatically this pathway is ignited. Then the levels of cytokinins and hormones and coenzymes, as well as the role of DNA is also important. So all these things play a major role. You can see this picture from the link given here in this slide. Just go through this. No need to by heart all these chemical structures, but have a glance. Okay. So we have seen that the first step is phosphoribosyl pyrophosphate formation, and then after that it starts and flows into at last inosine monophosphate. Once the inosine monophosphate is formed, after that it can be getting converted into other nitrogenous bases. So this pictorial representation of the complete pathway is a very catchy image, just because it is easy to remember that there are two glutamines here, and these orange spots are all ATPs. Okay, so you're getting where and all the ATP is getting involved, you can easily remember. So you just go through this image at least once, okay, and you can see the number of enzymes getting involved also. It will be very catchy. It'll be in your brain. It will never go from your brain. So just go through this once. From inosine monophosphate and getting an AMP as well as GMP is also, it's all linked. Means this is the precursor molecule for both these bases, and this is highly regulated by feedback inhibition. Okay, so that also we'll see. So this flow chart gives where and all it is being regulated. So all these reactions we can keep in mind if it is required, if you are writing for a biochemistry or kind of exam. So that will be very helpful. Just keep this picture in mind, then you will be remembering all these steps where and all it is getting regulated. Purines little bit, it is confusing for many people, but it is so simple because it started with ribose sugar, and after that everything is synthesized as a ribotide compound. So these ribotides actually means already they are having one ring structure. After that this ring structure is one more ring structure is added to this, and ring closer is the last step. So it is so simple to remember all these steps. You can easily go through this. Whereas pyrimidine synthesis is a less complex pathway when comparing the previous one. And here people say like it starts with the glutamine, but actually it starts with carbamyl phosphate. The carbamyl phosphate is actually formed from this glutamine, ATP, and one molecule of carbon dioxide. So all these things forms together and forms the carbamyl phosphate as the first compound in this pathway. And from this at last CPP is derived. So this catchy picture, you can just go through it once if you need, but many times you don't get very complex questions from these kind of pathways. So just go through how many molecules are involved. I said like this phosphoribosyl pyrophosphate is common for both these pathways. So in the purine biosynthesis also it was the first molecule, but here in pyrimidine biosynthesis it is getting involved in one of the reaction. This itself shows that somewhere the Chargaff rule is played in the pathway itself. Okay, means everywhere it is getting regulated. It means there is no loose stock, no wastage of energy in living organisms. It means everything is perfect. So it starts with the aspartate, and actually the carbamyl phosphate is the first compound which is being formed, and From this, actually, the pathway flows and at last CTP is produced. Okay. Please go through this summary once. It will help you in writing some of the questions if you get anything from this part. And at last, we know that all these bases are synthesized during this S phase. All of a sudden, we moved from biochemical pathway to mitosis and meiosis, cell division. Okay. So what you are seeing here is the GAP1 phase, synthesis phase, G2 phase, prophase, metaphase, anaphase, telophase, as well as the, again, G1 phase. It's a circle, actually, it is a linearized circle. Okay, just stretched in this one. Again, this is from your preferred reading textbook. You see this in the synthesis phase, cohesins are getting involved. These cohesins are a kind of structural maintenance protein present in the nucleus. Okay, so at last these cohesins get added to, means attached to this chromosome, and during this time all these synthesis of purines and pyrimidids happens, and then once everything is ready, all these purines, pyrimidids, whatever ingredients required for cooking a new replication cycle is ready, then this starts. So in this replication cycle, what happens is it is producing one more strand, and that strand is added, and up to this is in the synthesis phase. So in the synthesis phase itself, the complementary means a new strand is born in that stage. Then after that, the condensate plays a role, and at last the chromosomes gets condensed into a little bit smaller one than the prophase, so that something you can see in the microscope in the prophase. But it is all loosely packed. So though they are loosely packed, never these chromosomes get tangled together. So that means like all these chromosomes are placed apart, like they don't even overlap together unless it is forming chiasma in the later stage. So unless formation of chiasma, it is not at all crossing together. It means all these chromosomes are kept separate inside this prophase itself. And then again, condensate plays another round of action, and then a metaphase shortening of the chromosomes happens. Then surprise comes in action, and we stop up to this metaphase level in this session. Okay, so because the remaining steps we see in the cell division session, in the next 2 classes we will see that. So again, we are in the replication stage. You know that there are one leading strand, lagging strand. It runs in opposite ways, and but the synthesis happens in the same phase, and so that the lagging strand gets a Wakasaki fragment. So all these Wakasaki fragments are at last linked by Nick closer, and then a new strand is formed. Okay, so this happens inside this replication fork. So this we have seen in that one, that it is happening in the synthesis phase. I said in the previous session that in a chromatid, highly packed chromatid, there is only one double helix packed. Okay, there is nothing dumped together. So a single double helix is getting coiled, supercoiled, means it is all packed with the histone proteins, and it is all nucleosome pores, and everything is highly packed and coiled. At last we get a packed chromatid, and it is having, means normally each one will be having 2 chromatids. So there are 2 chromatids, and you get some alleles of the same gene in the same location, almost same place it will be present. So one of this will be from the paternal parent, and the next one is from the maternal parent. So the alleles will be different, can be different, maybe same also, means homozygous condition or heterozygous condition, it may be same or different. So once it is getting functional, means once the gene expression starts from one of these allele, there is no necessity for the other allele also getting expressed. So it depends on the gene regulation. It is not about which one is dominant, who is dominant, or recessive in sequence level. So the dominance and recessive, as somebody asked me today in the class, it is all related to the phenotypic expression. Okay, so gene expression is being regulated at multi-level, and it is expression of a single gene is not at all a role of a single gene. There are many other coding as well as non-coding genes getting involved in expression of a particular allele or a particular gene, whatever it is. Okay, so again, where we start is like cohesion comes, synthesis happens, and then replication, and then condensing. At last, you do have all these chromosomes. And here I said like the cohesion is a part of structural maintenance. Proteins and these SMC proteins are a group of proteins. Just go through this image once so that you will remember all the components easily. Okay, so this image is another catchy image for you to remember the cell cycle. Just see this image also once, store it in your brain as a pictorial memory, and this will easily help you in understanding the cell division. So we'll see cell division in the next class. The replication, actually we have seen the replication fork, but this replication happens in a discontinued way. At last, everything is getting connected, and at last sister chromatids are formed. So with this, I stop this session. We'll get back to you again in the next class. Thank you.